Hello everyone, and welcome to episode 35, Remastered. Today, I'll be covering the kingdom Animalia, which is a huge and incredibly complex kingdom that's very closely related to the fungi. Both animals and fungi are part of the clade Opisthokonta, which is one of the major subgroups of the Protista. I've been mentioning them several times over the last few episodes. But the animals and the fungus are categorized as their own groups, as their own kingdoms, because they're quite distinct in many ways, genetically, chemically, physiologically, morphologically, and so on. The animals are the kingdom that I want to explore today. It's going to be a really fun episode, because animals are simultaneously some of the most exotic and familiar organisms on the entire planet. You are an animal, as am I. As is everyone that we know. The animals live in the sea, they live on the ground, they fly in the air, and with a little help from rocket technology, they've even been in space. Understand that the story of animal evolution is also the story of our evolution, of how humans came to be. The story of the kingdom Animalia begins in the ages immediately preceding the Cambrian explosion, which began around 550 million years ago, and it lasted around 25 million years. Now, immediately before the Cambrian explosion, it was called the Ediacarian period, and during this time, animal life was very simple. The only animals that existed were sponges, and hydra, and jellyfish. Animal life was soft and simple, and it hadn't really even evolved bilateral symmetry yet. That would happen during the Cambrian explosion. This Cambrian explosion was like a veritable blossoming of life, where every single major animal phyla that exists today appeared. It's when they first appeared, including arthropods, mollusks, echinoderms, chordates, and a number of various phyla of worms. The Earth had been dominated by single-celled life and microbial colonies for billions of years. But the Cambrian explosion saw the rapid proliferation of multicellular organisms with increasingly complex body systems. Bacteria and archaea had long ago fused to create the first eukaryote, the first protista, and these diversified into numerous clades, which I talked about in episode 32. One of these clades of protista was the Opisthokonta, which includes the protists called choanoflagellates, which I've discussed in the last few episodes, as well as the fungus that I discussed in episode 34, and lastly, the animals. And the animals are the subject that I'm really excited to dig into today. Okay, so first, the basics. The most fundamental description of animals is that they are mobile heterotrophs. They're multicellular organisms that can move around and eat other organisms. Functionally, all animals have some kind of muscle cell that contorts and moves their body, as well as some kind of neural cell that sends signals to other cells. I should mention that sea sponges are the only exception to this, because they don't have nerves, or muscles, or organs, or really any kind of serious cellular specialization. They don't really have differentiated tissues. They're just really primitive. However, the sponges are still considered to be animals because they're multicellular heterotrophs that produce sperm cells and they don't have cell walls. Pretty much all animal cells do not possess a cell wall. The animal cells are soft and plastic. So fundamentally, animals are multicellular, mobile, chemical superstructures that eat non-stop, and they're able to move relatively easily compared to plants and fungus to acquire more food. This approach to life, this life strategy, allows animals to consume tremendous amounts of nutrients and chemical resources, which enables them to grow quite large and use all of that energy to power their movement. Where trees can grow tall because of the chemical lignin and the structural support that it provides, animals grow large because their mobility gives them access to an order of magnitude more nutrients. In all ecosystems, animals are the primary consumers of biomatter, the primary movers of chemical energy across the food chain. I think the only exception we can make to this is in the case of detritus, or dead biomatter. Because in that case, I think the primary consumers would be fungus, not animals. But when it comes to living stuff, or stuff that was just recently hunted down and killed minutes ago, that stuff is primarily consumed by animals. The very first multicellular animal was a sponge, from the phylum Porifera, which includes animals that filter feed through a network of pores. The sponges had existed for more than 50 million years before the Cambrian explosion, and their genetics and life strategy are, fundamentally, really similar to the choanoflagellate protista that they share a common ancestor with, which would suggest that they were one of the first diverging lineages from the protista. Or in other words, the sponges were among the first in a lineage that would become the animals. It's believed that at some point, a lineage of sponges evolved a little more complexity and eventually detached from the ocean floor, becoming the mobile, swimming organisms called the Ctenophora, or the comb jellies. These comb jellies have very simple bodies, composed of an outer layer defining their body shape and an inner layer defining their digestion cavity. You can see some patterns in this early evolution of the animal species right away. For example, the sponges, with their primitive, virtually undifferentiated tissues, have given evolutionary rise to the comb jellies, who have these two different differentiated layers of tissue, so we're slowly starting to see more complexity. Anyway, these comb jellies' bodies are lined with rows of cilia, little tiny hairs, and they rhythmically pulse these little hairs in order to move and swim through the water. The Ctenophora were also some of the first animals to evolve true nerve cells, but these nerve cells don't form any kind of centralized neural structure, like a brain or a spine. Instead, the comb jelly nerves form a decentralized mesh called a neural net that provides a unified sensory awareness across the entire body. This decentralized neural net is a trait that ctenophores share with the Cnidaria, which are the next major divergence in the animal lineage. The Cnidaria include the sea anemones, the corals, and the jellyfish, all organisms which possess a specific cell type called a cnidocyte. This cnidocyte is a rod or a bulb-shaped cell that literally explodes outwards in such a way so as to propel a small amount of toxin into another organism. It's like a poison bullet. The Cnidaria use these weaponized cells to deliver stings and paralyzing toxins, which come in handy when you're trying to capture and eat prey, especially when that prey can move faster than you. And it's also useful when you're trying to defend yourself from predators. These cnidocytes, these poison-launching cells, are present on the tentacles surrounding the Cnidarian's mouth, like the long tentacles on a jellyfish, or the thick, stubby tentacles on a sea anemone. Both the Ctenophoria and the Cnidaria are diploblast animals. This means that their body tissues are derived from an embryo that begins with just two distinct tissue types. These are the ectoderm, which covers the outside of the organism, and the endoderm, which lines the inside. For these two phyla, the Cnidaria and the Ctenophora, the ectoderm forms the soft skin that contains the jelly-like substance that's held within their bodies, while the endoderm becomes the inner layer that defines the digestive cavity. From the phylum Cnidaria diverged a relatively small phylum called the Acela, or the acoelomate worms. These worms shared traits like a decentralized neural net with their jellyfish and comb jelly ancestors, but they also possessed some novel evolutionary adaptations that would enable the development of larger, more complex life. The first of these important adaptations in the Acela was triploblasty, or the emergence of a third layer of germ cells in the embryo called a mesoderm. This mesoderm creates the specialized cells like muscles and vasculature and organs and all of these other structures that exist between the ectoderm and the endoderm. Now, the second important adaptation was the shift from radial symmetry that you would see in like a jellyfish or a coral to bilateral symmetry, which you would see in like a worm or in more complex organisms like fish and lizards and everything else. Where the jellyfish had a radial symmetry like a bicycle wheel or a clock, The ocella had a bilateral symmetry that was defined by a plane running down the length of their bodies, which divides them into mirror halves. This bilateral symmetry is the defining feature of all animal life that has diverged beyond the ocella, like the snails and squid, the insects, and vertebrates like fish and reptiles and amphibians, and everything else. In the evolutionary record, we're now around 500 to 450 million years ago, which is barely out of the Cambrian explosion, and we're about to witness the emergence of the triploblast bilaterians, a lineage of organisms with bilateral symmetry and an embryo with 3 differentiating layers of germ cells. These triploblast bilaterians are a huge group of animals, including virtually all animal life that's more complex than a jellyfish or an coelimate worm. Alright, well, let's look at the root of this lineage for a second. Let's examine the coelimate worms in a little more detail. The phylum Ocella literally means that they lack a coelom. But this just raises another question. What's a coelom? The answer is that it's one of the more important evolutionary emergences in the history of life. Consider all of the diploblast organisms like sponges and comb jellies and jellyfish. These organisms have 2 germ layers in their embryo, and these developed into the inner lining of the digestive tract and the outer lining that's the skin or the epidermis that's innervated with the nerves from this neural net. In diploblasts, these layers or linings are attached directly to one another, so the organisms take on a bag-like shape. Their bodies are just thin carbon fabric bags, which take in and excrete nutrients from the same opening. The triploblasts, in contrast, have that 3rd germ layer called a mesoderm, and it's positioned between the ectoderm and the endoderm. In the jellyfish and the sponges, their inner and outer tissue layers are directly connected. In bilaterians, the mesoderm separates them, and in organisms that have a coelom, the mesoderm itself contains pockets, or internal cavities. In coelomates, or in organisms with a true coelom, the mesoderm completely lines these cavities. Now, I know that this explanation might seem a little abstract, so consider it in real-world terms. Consider your own body. When you were an embryo, you had ectoderm, endoderm, and mesoderm germ cells. Your ectoderm developed into your skin and nerves. Your endoderm developed into your stomach and intestines, and the other tissues that line your gut. Your mesoderm forms the stuff between them, between your skin and your gut, or between your inner and outer linings. Your mesoderm develops into your muscles and your organs. And your organs sit within internal cavities. These internal cavities, or chambers that house your intestines and your heart and your lungs, all of these cavities are called coelom. The evolution of the coelom was really important because it created a fluid-filled cavity. This internal space could now easily transport nutrients throughout the body. It provided a space for organs, but most fundamentally, it turned soft-bodied creatures into pressurized capsules. With the internal pressure of liquid held within the coelom, the soft-bodied organisms enjoyed a kind of turgidity that acted like a hydrostatic skeleton. It's like the difference between an empty or a full water balloon. The water balloon has a stiffer surface that can apply greater pressure against the environment. The empty water balloon has none of this stiffness, it has none of this turgidity. It's soft and flaccid, and the environment can easily push it around. This is kind of like a soft-bodied organism. But in the case of the first coelomate organisms, this ability to apply greater pressure against the environment meant that the organisms could evolve limbs that could exert pressure on the water to help them swim, and because their bodies were a little stiffer, a little more solid, they could move more efficiently through the water. The evolution of the coelom greatly enhanced animal mobility, and when you can move quickly, you can get food more often, you can accumulate more food, and you can grow larger, and you can reproduce more. The coelom is also associated with 2 other important evolutionary adaptations. The neural net coalesced into a centralized nervous system, with a very primitive spinal cord. As an animal moves forward in the search for food, there was an evolutionary pressure for the nervous tissue at the front of the animal to become larger, with a greater density of nerves. In time, this increasing concentration of nerves became a very primitive brain, although at the time it would have been little more than a fleshy nodule called a cerebral ganglia. The development of this brain was part and parcel with the second major evolutionary adaptation, the development of a head. This process was called cephalization, and to put it simply, the bilaterian animals began to develop distinct head and body structures. At one end of the organism was a highly sensitive head that searched out and consumed food, and behind this head was a body that was evolving greater and greater mobility. The head and its dense nerve tissue detects stuff like food, or danger, or a potential mate, and the head will then send signals to the rest of the body. These signals are used to animate the body in such a way as to propel the head forward through the environment. This is a very important phenomenon to understand. Animals are very large chemical superstructures whose bodies are designed for two things: roaming around looking for food and eating that food. This is why pretty much all bilaterian species, from snakes and fish to cows and birds to humans, they all have heads on their front side, with a body replete with limbs or musculature that can transport the animal. The fins of a fish help propel and direct it through the water, as do the arms and the webbing of an octopus. Birds can walk around on their feet, but their upper limbs are their primary means of locomotion, as birds use their wings to fly and to maneuver through the air. Before I get too far into the way that animals move and eat, let me return to the subject of evolutionary history. Now we're at a point where the bilaterian animals have 3 germ layers in their embryos, and this is where the next major divergence would emerge. 2 general mechanisms for embryological development appeared, which would divide the animals into 2 very fundamental groups: the protostomes and the deuterostomes. Okay, so first, you should understand that the embryo is basically a spherical clump of cells, which are rapidly reproducing. The cells in each germ layer develop into the layers of the body, into the different tissues of the body. But the way these germ layers emerge and organize themselves physiologically differs between the 2 groups. It differs between protostomes and deuterostomes. The endoderm will bubble into the outer layer of the embryo to create a hole, which is either the exit or the entrance of the digestive tract, or the inner boundary layer of the animal that lines the internal cavity that's used to digest food. In protostomes, the endoderm pore creates the mouth first, and then a second pore forms, and this is the anus. And together, these 2 pores, connected by the internal cavity that is the gut, will form the digestive tract. But in deuterostomes, this process happens the other way. The first pore will become the anus, and the second pore that emerges will become the mouth. There's also a difference in how the mesoderm develops into the internal organs and muscle tissue. In protostomes, the mesoderm starts at first as a solid mass of cells, like a block. But these cells begin to hollow out from the inside to create a little internal pocket, or a bubble, which is entirely lined with mesoderm. The deuterostomes, on the other hand, have a mesoderm cell layer that's in contact with the endoderm, and when the embryo develops, the mesoderm cells will expand like bubbles that bud off of the endoderm. This expanding, bubble-like growth creates an empty internal space called the coelom that's also lined with mesoderm. Both protostomes and deuterostomes have the same end goal here during embryological development, which is to make a coelom that's entirely lined with mesoderm. They just have different ways of doing it. Evolutionarily, the protostomes would go on to evolve into organisms like flatworms, roundworms, earthworms, squid, snails, slugs, and other gastropods, and every single insect, arachnid, and crustacean on the planet. The deuterostomes would diverge into the starfish and other echinoderms, as well as all the animals with a vertebral column, like fish, amphibians, reptiles, birds, and mammals. In case you're wondering, I will be covering all of these groups in greater detail in future episodes. Now, as I've already discussed, the protostomes and deuterostomes had a common ancestor that possessed a coelom, a centralized nervous system that formed a rudimentary brain, and the emergence of a bilateral body symmetry that created a front-back body structure and a head. But beyond these similarities, the protostomes and deuterostomes diverged entirely, and their lineages have diverged through time into a bewilderingly diverse array of species. Here's a fun little detail. The protostome group, called the platyhelminths, or just the flatworms, actually lost their coelom, as their mesoderm gradually evolved smaller and smaller internal spaces until the mesoderm was more or less a solid block of cells again. In other groups, the mature adult body form evolved again a kind of full or partial radial symmetry, like the deuterostome starfish in the phylum Echinodermata, or like species in the mollusk phylum, like squid and octopus. There were 3 major bilaterian phyla that independently evolved segmented bodies, or some kind of segmentation. The annelids are one such phyla, which are literally called the segmented worms. The other phyla that evolved segmentation are the arthropods and the chordates, so pretty much all of the insects, all of the crustaceans, and all of the vertebrates. Segmentation involves the presence of repeating structures or subunits in the body. The bones in your spine are an example of segmentation, as are the body sections and jointed limbs of a spider, a crab, or a dragonfly. If you listen to the episodes in my series on genetics, you might recall that genes are often expressed in groups. Any single given gene produces an enzyme, or part of an enzyme, and these work in tandem with a large group of other enzymes to perform some kind of greater function or utility. You can think of these enzymes and the genes that code for them kind of like the tools in a toolkit. The tools you have in a given toolkit will vary depending on the job at hand. For example, a carpenter's toolkit will contain stuff like a hammer, nails, a ruler, a level, that kind of thing. While a road engineer would have similar but slightly different tools, like a ruler and a level, but a flat striker bar instead of nails, and a sieve stack to analyze gravel instead of a hammer. Now, in much the same way, groups of genes that code for enzymes that need to work together are typically all expressed at the same time, with slight variations for specific purposes. These gene groups behave like a toolkit, printing out groups of enzymes that all work together in particular ways to perform specific functions. If there are small changes in the genes that are being expressed down the length of the animal's body, the animal will develop differences in the size, shape, and even the number of its limbs, as well as the segmentation and organization of its body structure. This segmentation is the product of a streamlined system of genetic expression, which modifies genes as needed to create the animal's anatomy. For example, your thumb and your other fingers are very similar structures, but they obviously have differences. The thumb is shorter, it has one less joint, and it's positioned at a different angle than the other fingers. But the genes that produced both your thumb and the rest of your fingers are very similar, with only slight modifications in expression. Those modifications are the result of evolution shaping our physiology, and they're responsible for the anatomical and physiological differences that I mentioned. In much the same way, the groups of genes that are used to create your toes are very similar to the genes used to create your fingers, but there's differences in how these genes are expressed that creates the anatomical differences between your toes and your fingers. For example, your toes are shorter and stubbier, and they're much less dexterous. Similarly, segmentation and slightly variable gene expression occurs in your finger bones themselves, which get smaller as they get closer to your fingertips. And segmentation also applies to your vertebra, which get larger as they go down from cervical to lumbar. It's also why some animals have multiple pairs of legs, with one pair often much larger than the others, like the jumping legs on a grasshopper or a frog, or the claws on a crab. When you're looking at a myriapod, like a centipede or a millipede, or any other animal that has a large number of very similar repeating segments, these repeating segments are generated by the same groups of genes being expressed over and over and over again. And each time this group of genes is expressed, it creates another body segment. Okay, so now that I've touched on the major parts of animal evolution, let's explore what behavioral motives and selection pressures were behind all of this rapid diversification. I've established that animals are eating machines, basically just massive chemical superstructures that move across the environment and eat. So how have they evolved to move? And how have they evolved to eat? Well, movement is achieved by moving their limbs in coordinated ways, which propels the head and the body through the environment, be it water or air or dry land. In all animals, the limb develops as a stub, or a little outgrowth of cells. A gene called DLL is heavily expressed in this early limb nub, and it directs the growth of the cells outwards, extending the limb from the developing body. Some animals have little more than these rudimentary stubs or lobes, which they use to crawl around. Other animals have tentacles, while others still have jointed limbs. You're probably most familiar with jointed limbs, because you have them, as do all the other vertebrates and all the arthropods. We use our jointed limbs to move, to walk and run and jump, or to climb and swing through trees. Humans use their jointed arms and hands to throw spears, and carve up the meat from the animals that they hunted. Speaking of eating meat, this is a nice transition into the methods that animals use to eat. You're probably very familiar with what animals eat. Carnivores are predatory animals that eat other animals. Then you have the less dangerous herbivores that eat the plants. And then you have the detrivores that eat dead and decaying organic matter. Some animals are able to eat both other animals and plants, and these are called omnivores. They include some species of bears, ants, birds, and of course, humans. Humans are omnivorous. Now, it's pretty important to understand that what an animal eats plays a huge part in how that animal species is involved in its local ecology. The ecology is the net sum of the interactions between organisms and their physical environment, and how those interactions move resources and nutrients through geographic space. The food chain is a very simple ecological diagram, showing plants as the primary producers, which get eaten by the herbivores. The plants will turn light energy from the sun into chemical energy in the form of sugars and biomolecules, and when herbivores eat the plants, they eat the biomolecules, and they harvest a portion of the total chemical energy that exists in the plant. When a carnivorous predator captures and eats the herbivore, the chemical energy in the herbivore's body will get digested by the predator, and the predator will harvest a portion of that chemical energy for itself. Because of the reduced amount of chemical energy that's available in a stable ecosystem as you go up and up these layers of the food chain, you start to see that predator populations are often much smaller than prey populations. When there's no predators at all, the population of prey animals, or herbivores, generally skyrockets. This swollen population of herbivores will eat all of the available food, and then they'll begin to starve. But if predators were to come into the habitat and see this swollen herbivore population, then the predator population can also swell, because their prey has increased in number. But when the prey starts to starve, because they ate all the available plants, the engorged predator population will also start to run out of food, and it too will experience starvation. In this way, the population sizes of associate predator and prey species often goes through a cycle, where the growth or the decline of a predator population follows at a slight delay after the prey population grows or declines. Okay, so that's what animals eat, but how do they eat? Conveniently enough, There are 4 primary feeding strategies that animals use: filter feeding, deposit feeding, those who consume fluids or fluid feeding, and those who consume raw chunks of flesh, or the mass feeders. The filter feeding animals are hugely diverse, including the small and stationary sponges, which filter food particles out of the water, and baleen whales, which take in a huge mouthful of ocean water and squeeze it out, filtering out the water through its baleen teeth, but capturing plankton and krill for food. The deposit feeders aren't as diverse, as they're usually some kind of worm or worm-like shape. These deposit feeders will dig through the substrate, be it the sand on the seafloor or the dirt on dry land, and they'll digest organic particulates and microbes and other ground-dwelling organisms. Their excretion will go back into the soil and replenish it with nutrients. The fluid feeders are like the opposite of filter feeders. Where the filter feeders take in a huge amount of water and then filter it out for a bit of food, the fluid feeders typically use a very thin proboscis to siphon up liquid. Butterflies and hummingbirds, for example, are fluid feeders that get their nectar from deep inside a flower. Other fluid feeders include bats and parasites that consume blood, stealing the digested nutrients straight out of another organism's body. The 4th feeding strategy is called mass feeding, and it literally involves an animal just taking a mass of food into its mouth and ripping it off of its prey's body and then processing it into a digestible format, usually through chewing with the teeth and chemical softening with saliva. These mass feeders include animals like cows and deer, as well as wolves and lions, and us humans. Us humans are also mass feeders. On a side note, recognize that all of these animals have evolved teeth shaped by evolution to specialize at consuming what they eat. Herbivores have broad, flat teeth and a lateral chewing motion that grinds up leaves. Carnivores have sharp teeth that can slash, puncture, and tear at flesh. The last thing that I want to cover today is perhaps the most fundamental. The animals may be the only organisms that move around to seek out food. I mean, the fungus might be a technical exception, but everything needs to reproduce, and animals are obviously no exception. The purpose of the organism moving around is to find food to eat, and the whole purpose of finding food to eat is to stay alive and grow big and healthy. The whole point of growing big and healthy from an evolutionary perspective is to increase your chances at having sex, at reproducing and passing on your genes. Some animals are asexual, like the corals, which reproduce by forming little polyps that bud off of the parent's body and grow into new individuals. Asexual reproduction is relatively efficient because it just requires one individual. And if the reproduction process isn't fatal, that individual can reproduce on their own over and over and over again. But this asexual individual is only producing clones, and a big population of clones has very little genetic diversity. Low genetic diversity makes the asexual populations vulnerable to diseases and other large-scale environmental threats. Sexual reproduction evolved to solve this problem, as sexual reproduction involves the mixing of 2 different individuals' genomes into an offspring with new combinations of alleles, and thus, novel genetic variety. Sexual reproduction can be either internal or external, and this is a reference to where the fertilization takes place. Animals like fish and sea cucumbers use external fertilization, where they spray their eggs and their sperm into the water. The sperm fertilizes the eggs, and the embryo develops outside the body of either parent. With internal fertilization, the male sperm is deposited through a variety of ways into the female's body, where it fertilizes her eggs. Once the egg inside the female's body has been fertilized, there are several ways that the embryo can develop, depending on the species. You have viviparous animals, which grow the embryo inside their bodies, nourishing it with their own blood until they give birth to live young. Humans are viviparous, as are cats and dogs and most other mammal species. But then there are the oviparous species, which remove the embryo after fertilization. The embryo is grown inside a protective sac called an egg, and it's deposited somewhere outside the parent's body. For example, insects will lay their eggs on the underside of leaves, or in a piece of fruit, or even inside the body of another animal. The reptiles will lay their eggs and then bury them in the sand or the dirt for safekeeping. The birds will lay hard-shelled eggs that they keep in a protective nest, and they tend to watch over them until the eggs hatch. There's also ovoviviparous species, which are a strange combination, retaining their eggs internally but giving birth to live young, so the eggs hatch inside of the mother's body. However they're born, the young offspring will go through a life cycle, maturing through adolescence until they reach adulthood, where they'll reproduce and have offspring of their own. Some animal species die immediately after reproduction, but others can live on and have more offspring before they die. Those offspring will grow and reproduce and die, and their offspring will grow and reproduce and die, and this will go on and on across evolutionary time. This is how genes and alleles and entire species are propagated through millions of years. Alright, that's all that I have for you today. If you want to hear more stuff about animals, just know that I'll be creating future series on both the protostomes and the deuterostomes, and these episodes will explore their evolution and their physiology in much greater detail. If you enjoyed this episode, please give it a like, and if you like all my content, then subscribe to my channel so you can keep up with the new stuff that's released every week. If you want to support the podcast in other ways, consider shopping around on the official store with stuff that I personally designed myself. Or if you want to support the show in other ways, consider becoming a patron. Anything you do supports the podcast, and I greatly appreciate it. And as always, thanks for listening.